Use the image hydrophobic barrier pen to apply a waxy barrier to the slide. This barrier will localize the antibody solutions to the section, allowing you to use less solution per section, or to use different antibody solutions on different sections on the same slide. If you are using the pen for the first time, give the pen a good shake. Depress the nib into the pen by pressing down on the pen tip on a soft surface like a couple of paper towels. The blue solution will fill the nib and the pen will be ready for use. Use the pen to draw a line between tissue sections on a slide or to draw circles around each section on the slide. Use the imprint pen to label the slides. The imprint pen is resistant to most organic solvents like alcohols and clearing agents that are commonly used in staining protocols. The ImmPRESS reagent is a polymer-based detection system that consists of a polymer of peroxidase directly conjugated to the secondary antibody. Each ImmPRESS kit contains two components, a pre-diluted normal serum for blocking and the polymer detection reagent in a ready-to-use solution. ImmPRESS reagents are available to detect mouse, rabbit, or goat IgG. A universal reagent is also available with dual specificity for mouse and rabbit IgG. Because of its convenient format, the ImmPRESS reagent can significantly shorten multiple labeling protocols. The reagent provides very high sensitivity, achieving similar sensitivity to the Vector Elite ABC kits with very low background. The system is also biotin-free, which is ideal for those applications where endogenous biotin is present. The normal horse serum serves as a general protein blocking reagent to minimize nonspecific interactions of subsequent detection reagents. Remove the slides from the buffer and briefly wipe the excess buffer with a tissue. Cover the sections with diluted normal horse serum by applying several drops of serum straight from the bottle onto the tissue. Incubate for 20 minutes in a moistened, darkened chamber. Dilute the primary antibody. Optimal dilution needs to be determined. If your antibody is commercially available, a recommended range may be supplied. Tip off the excess serum from the sections onto a stack of paper towels as shown. Cover the section with the diluted primary antibody solution. The optimal incubation times needs to be determined. A recommended range may be supplied by the antibody manufacturer. Rinse the sections and place the slides in a Copland jar for 5 minutes. Blot the buffer from the sections. Cover the sections with the ImmPRESS reagent by dropping several drops of solution from the bottle directly on the tissue. Incubate for 30 minutes. Rinse the slides with buffer, then place the slides in a Copland jar with buffer for at least 5 minutes.